Hello students, today we are going to discuss two topics, that is vectors for cloning genes in plants and animals, and second topic we will discuss is about competent host. In previous lectures we have learned about biotechnological tools in which we have learned about restriction endonuclease enzyme, which are the enzymes used for the cutting the DNA molecule, and that's why it is also called as a biological scissor or a nuclear scissor, and it is very important tool to cut the DNA from specific region, and in the biotechnology this cutting of the DNA is playing very important role. Second thing which we have already discussed is about cloning vectors, which are the tool which are used as a we can say as a tool for the transporting DNA from one organism to another. And today we are going to discuss about some processes by using which we can enter this cloning vector into the host. Before we start the topic competent host, let me discuss about some vector which are used for the cloning, okay, and for the cloning genes into the plant cells and animal cell. So genetic tools of some pathogen is transformed into the useful vector. We can modify this pathogen, though they are pathogen, we can transform them and we can use them as a vector for delivering genes to plant cell and animal cell. Such an example is Agrobacterium tumefaciens, that is a one kind of bacteria. Normally it is a pathogen of many dicot plant, but we can modify that genes, okay, and we can use them as a vector. Second organism is one kind of virus, and that is a retrovirus. We have learned already how much this retrovirus can be the pathogenic. Okay, we have learned already about HIV virus. HIV virus is one kind of a retrovirus. So we can modify these virus also for the our purpose. Okay, and we can use them as a useful. We discuss in detail about Agrobacterium tumefaciens bacteria, which can deliver a piece of a DNA, that is a tDNA. And why it is written tDNA? Because T for a tumor. Okay, so this DNA is responsible for causing tumor, and it will transform normal plant cell into the tumor cell. Okay, so that's why this DNA is considered as a tDNA. So these tumor cells produce the chemicals required by the pathogens. Okay, so by this way, Agrobacterium tumefaciens can deliver a piece of a DNA, that is a tDNA, and which is responsible for causing tumors in the plant cell. Now, tumor-inducing plasmid, that is also called TI plasmid, TI, that is a tumor-inducing plasmid of Agrobacterium tumefaciens, is modified into a cloning vector. So we can modify it and we can enter it into the cloning vector, which is not pathogenic but can also use a mechanism to deliver a genes. Okay, it is actually not pathogenic. We have modified it, but it can use as a mechanism to deliver the genes of interest into the plant cell. So here you can see we have inserted a tDNA into the TI plasmid. This is a TI plasmid, that is a tumor inducing plasmid, in which we have transferred this, means we have attached this tDNA. Okay, now these TI plasmid is not responsible for causing any disease. Okay, and we can transfer, means by using this mechanism we can deliver genes of interest into the next pathogenic organism which is used as a vector is a retrovirus, and these retrovirus is mainly used for the transfer of genes in the animals. Retroviruses in animals can transform normal cell into the cancerous cell. Okay, so we have previously discussed about Agrobacterium tumefaciens, and that is responsible for causing normal cells into the tumor cells. But here you can see retroviruses have the ability to convert or transfer normal cell into the cancerous cell. So they are used to deliver desirable genes into the animal cell. So here you can see this is the cell, normal cell. Okay, retrovirus will be infect here, so that gene will be incorporated into the host DNA, and by this way this cell will now convert into cancerous cell. So retrovirus here play very important role if we convert them into non-infectious or non-pathogenic. Now we are going to discuss about third tool of a biotechnology, and that is competent host. So why this competent host is important? Because we have to now enter these recombined plasmid into the host cell, and for the entry of the recombinant plasmid inside the host cell, these theory play very important role. So let me understand. Since DNA is a hydrophilic molecule, you have learned already in standard 11 chapter 9 that DNA is a negatively charged molecule, and that's why it is hydrophilic molecule. It cannot pass through cell membrane. Why DNA cannot pass through cell membrane? Because cell membrane itself is negatively charged. So here what happened? DNA itself is a negatively charged and cell membrane itself is a negatively charged. So both the charge will be negative, and that's why these plasmid cannot enter or pass from this cell membrane of a host, and that's why we have to make the charge into, means we have to change the charge to entry inside the host cell. So how we can do this? Let me try to understand. So the bacterial cells must be made competent to take up the alien DNA or a plasmid. Okay, for these what we are doing? Bacterial cells are treated with a specific concentration of a divalent cation, that is, for example, we can say a calcium. Okay, calcium you know very well. Calcium is a positively charged. So if you treat the bacteria with the calcium, or you can see here calcium chloride treatment, you giving the bacteria the calcium chloride treatment, what happens? The cell surface or a cell membrane will be covered with the calcium ions, and as a result of that it will convert into positively charged. Okay, now what happened? This DNA molecule, this is a plasmid that will be negative in charge, and these will become, after calcium treatment, it become positive charge. So that's why these plasmid will now easily enter inside the host cell. So calcium treatment play very important role, and after that you have to give the heat shock, and that is 42 degrees Celsius for the 2 minutes, and that why now you can see plasmid will be transported inside the cell, and you can see here this is the bacteria which is transformed. Okay, otherwise it is not possible to transform the bacteria. So DNA enters the bacterium through the pores in the cell wall. During recombinant DNA technology, it is very important step that recombined plasmid must be enter inside the host cell. But during the entry inside the host cell, DNA or a plasmid will be prohibited due to two reasons. Because DNA itself is a negatively charged, and second thing, DNA itself is a macromolecule, large size molecule. That's why it cannot directly enter inside the host cell, and for that we have to make them competent by using several methods, and such a method include microinjection. Okay, so how we can do this? So here you can see this is a microinjection in which this recombinant DNA will be filled and then directly it will be injected in the nucleus. Okay, so recombinant DNA is directly injected into the nucleus of an animal cell by using microinjection. Okay, second method is a biolistic method. It is also called gene gun. Cells are bombarded with the high velocity microparticles, and that microparticle mostly used is a gold and tungsten. So we can cover or coat the DNA with the help of a microparticle, that is gold and tungsten, and after that we can enter inside the host cell. This method is suitable for the plant cell. Third method is a disarm pathogen vector when it is infect the cell. Okay, so it is a generally such a pathogen, means these are the, normally they are pathogen, but we can disarm them, means we can inactivate them as a pathogen, and after that we can infect the cell, okay, which is less harmful now, and we can transfer the recombinant DNA into the host cell. Another important method of a competent host is a lipofaction. Okay, so by using lipofaction also we can make the host competent. So here you can see plasmid DNA is there. It is covered with the lipid. Okay, there is a transfection reagent. Okay, lipid along with some reagent. It will cover, and now this DNA can enter inside the cells, host cells. That is a lipofaction method. Now let me understand more by using this diagram about the microinjection. So here you can see this is the plasmid DNA which is filled inside this microinjection, and by using this microinjection it is given injected directly inside the nucleus. So by this way this plasmid DNA, which is recombined plasmid DNA, will be inside, means enter inside the nucleus directly. Next is a gene gun method. Here you can see some synonyms of the gene gun method, that is a particle gun, particle bombardment method, biolistic method, microprojectile method. So these are all are the synonyms of the gene gun method. Okay, so in which here you can see this is a gene gun. Okay, in which DNA will be filled. There is a plastic disc with the DNA coated gold particle, sometime in tungsten particle also used. Okay, this topped by the screen, and then DNA coated the gold particle will be bombarded on the cells, that is a target plant cell, and now this DNA coated gold particle will be enter inside the target cell plants. So by this way we can avoid the rejection. This is your homework for today's lecture, and before we finish this lecture, let me summarize whole topic by using one animation video. So look at this video.
Interestingly, human beings have learnt about cloning vectors and the concept of gene transfer into plants and animals from bacteria and viruses. Consider the case of Agrobacterium tumefaciens, a pathogen of several dicot plants which causes tumors in them. It delivers a piece of DNA known as tDNA and transforms normal plant cells into tumor cells. These tumor cells are used by the pathogen to produce chemicals such as naupoline required by it. By understanding how pathogens deliver genes in their hosts, scientists have been able to transform the pathogen into useful cloning vectors. For instance, the tumor-inducing Agrobacterium tumefaciens has been modified into a cloning vector Ti plasmid, which is not pathogenic to plants. In fact, the Ti plasmid vector uses the same mechanism to deliver other genes of interest in plants. Once we have obtained the recombinant DNA molecule using a cloning vector, another challenge is introducing this recombinant DNA in the host cells. In DNA, the deoxyribose sugar and the phosphates are hydrophilic in nature, therefore cannot pass through the cell membranes. To overcome this problem, bacterial cells have to be made competent to take up plasmids. Therefore, they are treated with divalent cation like calcium. This enhances the efficiency of the entry of DNA into a bacterium through pores on the cell wall. After this, recombinant DNA can be forced into such cells by incubation of cells with recombinant DNA on ice. This process is followed by administering a heat shock to the cells at around 42 degrees Celsius and putting them back on ice. After this, the bacteria are able to take up the recombinant DNA. Apart from cloning vectors, foreign DNA fragments can also be introduced into host cells using methods such as microinjection and biolistics or a gene gun. The microinjection method involves the direct injection of recombinant DNA into the nucleus of an animal cell. While in the biolistics or gene gun method, plant cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA. This method uses disarmed pathogen vectors, which, when allowed to infect the cell, transfer the recombinant DNA into the host. The use of cloning vectors and methods such as microinjection and biolistics are crucial in genetic engineering as they help create genes of human and scientific interest.
In next lecture, we will learn about processes of recombinant DNA technology. So till that, goodbye.